You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is September 27th, 2024. We will be doing How the Immune System Works by Dr. Lauren Sompayrac. We will be discussing chapters 8 through 12. This is presented by Dr. Nikita Raje. She is an associate professor of internal medicine pediatrics and is the chief in the section of allergy immunology at Children's Mercy Hospital in Kansas City, Missouri.
It will be a shorter discussion. We have several chapters to go through, but really, it's just kind of moving through one through to another. We'll go from chapters 8 to 12 today, and then we'll continue with the rest of the chapters 13 to 17 next time. Let's see if we can move this here. We know we are going through basic immunology coursework. Our current textbook is How the Immune System Works with Lauren Sompayrac, and I'm Nikita Raje with Children's Mercy, University of Missouri, Kansas City. And today is September 27, 2024. All right, so here are my disclosures, nothing relevant to the talk today, and here are the learning objectives. We want to have more understanding about the restraining of the immune system, self-tolerance and MNC restriction, immunological memory, intestinal immune system, and then immune system gone wrong. So these are the 5 chapters we are going to go through. The first one is restraining the immune system. So we talked about activation and we talked about the effector system or adaptive immune system. We have talked about innate immune system, but we understand that the immune system has a lot of power. It can actually help protect our body, but also it has too much power, so that if unrestricted, it can actually cause damage. So we know that we need some counter processes in place so that the immune system can be attenuated. So there are several in place, and one of them is how the immune response is attenuated by inducible Tregs. So iTregs are separate from the natural or thymic Tregs that we know of. So iTregs are inducible Tregs, or they are also known as peripheral Tregs. They are found in, for example, in intestine, and we know that the T cells have various subsets, and under the influence of various cytokines, they decide to become particular type of subset, then releases cytokines that are helpful under that situation. So in terms of Tregs, if the antigen presenting cell presents an antigen in presence of cytokine of TGF-beta environment, then the Th0 cells are induced to become iTregs. And these iTregs then release TGF-beta or IL-10. These are the 2 anti-inflammatory cytokines. So whenever you are asked about anti-inflammatory cytokines, these are the 2 names that you need to think of: TGF-beta and IL-10. So what do these cytokines do? TGF-beta helps in reduction of T cell proliferation, and then they make the CTLs less vicious killers. IL-10 blocks the co-stimulatory signals, so it is difficult to activate naive T cells. So they help to reduce the activation, help to keep the activated T cells to be less active or less vicious, and help to calm down the immune response. So that's really nice, but what if the immune system's already activated? How does the immune response calm down and we reach our homeostasis back? And so there are several steps. For the first, it's just the reduction of antigen that helps in calming down the immune system naturally. Why does that happen? Because once the immune system controls the infection, say there is flu virus, my favorite example. So if there is a flu virus and the flu is cleared, the symptoms are gone, the infection is cleared, the that invader is destroyed, which means the antigens that were present, that were initially activating the immune system, are gone. And so if there is less antigen present, there is less activation of the innate immune system, such as antigen presenting cells are less activated. The sentinel cells are less activated. Now, we learned that the innate immune system is the one that activates the adaptive immune system. So if the innate immune system is less activated, it's going to not be as active in letting the adaptive immune system know that there is an infection. So overall, naturally, as the infection goes down, the antigen is reduced, the innate and adaptive system are less activated. But the weapons that are already formed, the cells or proteins that are already active, are going to be there, and they're going to continue to do their job. Thankfully, most of these weapons have a short half-life, such as neutrophils just live for a few days, natural killer cells live for a week, macrophages are typically in a resting stage. So once they are activated, they'll do their job, but then they'll go back to their resting stage. And then the dendritic cells live for a week or so. Plasma B cells that produce antibodies only live for a few days as well. And then the antibodies that are produced have a very short half-life. IgA, IgM, all the IgGs, other than IgG3, that all of them have, even IgG3 actually has the shortest half-life. But overall, IgG, immunoglobulin G, has the longest half-life among all antibodies, and that too is 3 weeks, right? 3 to 4 weeks. So over a period of few days, most of these weapons that were formed are gone. But then there are cells such as T cells that can stay activated for a while. So it's important to have checkpoints for these T cells. And so to deactivate the T cells, there are these checkpoint proteins, are what they are called, to decommission the T cells that are already activated. And they are called CTLA4 and PD-1. So what is CTLA4? CTLA4, so remember when we talked about antigen presenting cell interacting with a naive T cell for activation, it needs that second signal. That second signal is provided by CD28 interacting with B7 proteins, right? Now, the B7 protein is important to be able to activate the naive T cells, but when that activated T cell has been there for a little bit, instead of expressing a lot of CD28, even though CD28 continues to be on the surface, there is a lot more CTLA4 on the surface. And CTLA4 also interacts with the same protein as CD28. So there is competition between them. And CTLA4 has 1,000 times more affinity for those B7 proteins. So who wins? It's CTLA4. So when CTLA4 is present, CD28 cannot give it a competition. But thankfully, when it's just the naive T cells around, for activation of naive T cells, there is a lot of CD28 that is used so that that activation can occur. Once it's activated for a while, CD28 is overtaken by that CTLA4, which will now interact with B7. So let's look at what B7 does. CTLA4 is present in the naive T cells, but it's intracellular. Once activated, it gets on the cell surface, and then once that happens, once that interaction happens, it provides competition to that CD28. CD28 is activating, and CTLA4 is inhibitory. So it's more difficult to reactivate those T cells once they interact. Once there is interaction between B7 proteins with CTLA4. Also, there is a protein called PD-1 on activated T cells. What happens is some of the inflamed cells can express PD-1 ligand on their surface, and when that ligand interacts with PD-1 on the activated T cells, it prevents the proliferation of those activated cells, which means it's also a mechanism to deactivate those T cells. One more thing to remember is, say they are already activated. fine. CTLA4 can prevent that activation. PD1 can prevent that activation. But what happens to the T cells that are already activated? So thankfully, there is something called AICD, so activation-induced cell death, or also known as T cell exhaustion, which means that when the T cells have been have stayed active for a while, they have been fighting these infections, and basically they are just tired, right? So those T cells over a period of time will express, for example, they will express FAS FAS protein, and when that happens, there are CTLs that express FAS ligand on them, so they can bind to those, and because of that, they'll undergo cell death. So that's called activation-induced cell death. The naive T cells, why do they not undergo AICD? It's because they are insensitive to binding to FAS ligand, whereas the activated T cells then become sensitive to that FAS ligand, and because of that, they will undergo AICD. All right, so that's how the immune system is restricted in its function. Now we want to kind of backtrack and talk about the development of tolerance before the immune system goes into action. So we are backtracking and seeing how is the tolerance for the immune system is built. So there are several layers of tolerance, and it's important to understand that this is really, really important for our body, so that there are no self-reactive immune cells that start attacking its own body. So while the immunes, we talked about all stages of the maturation, and then of the T cells, B cells, how the innate immune system develops. We talked about the activation and the effects of it, and how it can, the inflammation or the immune system can be restricted. However, in the first place, if it's a self-antigen against which this immune system becomes active, that can be a problem. So to prevent that, there are certain layers of tolerance built into the immune system so that the immune system doesn't attack our own body. So that's central tolerance. There are some traffic patterns and secondary lymphoid organ in place which help in preventing the immune system being self-reactive. Sometimes there are missing signals, and then activation-induced apoptosis. So we'll look at each of these layers. So central tolerance. So we know that central tolerance is it comes into effect in thymus. Thymus is our primary lymphoid organ. Its structure has cortex and medulla. It has no incoming lymphatics. And we really don't know exactly how the T cell progenitors get into the thymus. There are no high endothelial venules either, but there is restricted entry of cells from the bone marrow that are destined to become T cells. And these cells, once they develop and mature, they exit near the corticomedullary junction into the blood. So let's look at how that progression occurs. So once the cells from the bone marrow that are T cell progenitors enter the cortex of the thymus, at that time, they do not have any markers that define them as T cells. They are still lymphoid progenitors that are destined to become T cells. When they go through that development of proliferating in that cortex, they eventually start expressing CD3 on their surface. As they express CD3, they become the T cells. T cells are defined by a presence of CD3 on their surface. And then besides the CD3, they'll start expressing both CD4 and CD8 co-receptors on them. So they are double positive T cells. Once those double positive T cells come in contact with the cortical thymic epithelial cell, or CTECs, TECs, right? CTECs, they undergo a process called positive deselection, where they are asked, can you interact with the MHC on our surface? And if the answer is yes, those double positive T cells survive. If not, they die, right? So they undergo autophagy, and there are only few who survive. Once they go through positive selection, they move to the medulla, and there they are asked a question. These cells are put under a test, and they are asked a question of, can you interact with MHC class 1 associated antigen or not. Class 1 or class 2 antigen associated MHC or not. And the correct answer is no. If they cannot interact with a self antigen, then those cells will survive. If they are self-reactive T cells that can interact with the antigen expressed by the thymic epithelial cells, medullary thymic epithelial cells, or thymic dendritic cells, in those cases, those single positive cells will die. If they interact with class 1 MHC, they become CD8 single positive cells. If they interact with MHC class 2, they become CD4 positive single positive cells. And those single positive cells are now ready to move out of the thymus. So they are called thymic emigrants. So we are talking about the same process here, but as you see, some of the markers on these change. So initially, the cells that are destined to become T cells enter the thymic cortex. They do not have fast antigen on them. They have high apoptotic proteins, because of which they are prone to proliferate a lot, and then over time a lot of these cells that do not express the correct markers, or when they undergo positive and negative selection, a lot of these cells die. So initially the double positive T cells might be about 60 million, but as they go through positive selection and negative selection, if they pass those tests, they graduate and come out of the thymus, and only about 2 million will pass both these tests. So they undergo this testing for over a period of 2 weeks. Where they are tested against the cortical thymic epithelial cells and medullary thymic epithelial cells to see if they can survive. That's the central tolerance. But once the thymic emigrants or recent thymic emigrants recirculate from one secondary lymphoid organ to another, they are called naive T cells. And these naive T cells only go from secondary lymphoid organ from one to another. They are not going into the tissues. So the abundant self-antigens in the secondary lymphoid organs are likely that were already present in thymus, and the autoreactive T cells were likely eliminated in thymus during that positive negative selection itself. If it's a rare self-antigen that wasn't presented to it in thymus, it's also rare in the secondary lymphoid organ. So likely it's not going to come across that antigen, or the antigen will not be enough to cross-link several T cell receptors. So this is called tolerance by ignorance. It's just the traffic pattern as well as the amount of self-antigen that's present that it gets ignored by the immune system. Another way of inducing tolerance in secondary lymphoid organs is presence of natural Tregs. So we talked about the inducible Tregs. Now we are talking about the natural or central or thymic Tregs, all different names for the same thing. These are CD4 positive, FOXP3 positive cells. They enter the lymph nodes and secondary lymphoid and other secondary lymphoid organs and protect against the T cells being activated if they are able to bind to a self-antigen. Peripheral tolerance induction, if these T cells that are self-reactive, they underwent that second positive and negative and positive selection in thymus and still were able to get to the secondary lymphoid organ. They went through secondary lymphoid organ and they still got activated. If they escape into the tissues, the self-antigens on the tissue cells may bind to the naive T cells, but because it's not a danger, it's not a foreign body, there is no co-stimulatory molecule. So there is no second signal for these T cells to get activated in large amount. So even though there is that risk of mild activation, they do not get activated as you would expect with another antigen. And this process is called energy. And then another one is called tolerance due to AICD, which means if it's a self-reactive naive T cell, it escapes thymus, enters the tissue, finds the antigen, maybe even find co-stimulation by any chance. Those T cells over time are going to get exhausted and undergo AICD. Apart from T cell tolerance, there is B cell tolerance, pretty similar. It also uses the ignorance and energy that we talked about with T cells. And however, there are a couple of other things that help with B cell tolerance. During the recombination, if there is a self-reactive B cell that can bind to self-antigen, the bone during that their development, these B cells actually, instead of killing that B cell, will provide a second chance to the B cells to undergo receptor editing, so that the self-reactive B cell gets changed to some that they can react to some other antigen rather than a self-antigen. Also, another process is where in the germinal center, when they undergo somatic hypermutation, a self-reacting B cell will not come across the antigen that is expressed to them in the germinal center by the follicular dendritic cells, because of which there is no co-stimulation for T follicular helper cells, and so those B cells will not proliferate as much. So they will eventually not be there. There might not be as many self-reactive B cells. Apart from B cells, the NK cells also have some positive selection. So we need to make sure that even the innate cells actually are not self-reactive. And so for NK cells, there are inhibitory receptors on their surface that interact with class 1 MHC. And if they do not detect class 1 MHC on the surface, they will kill the target. But if there is missing self-recognition, which means that if the class 1 MHC is missing on their surface, then they kill that target. So NK cells that do not recognize self-MHC are not going to be functional. So for them to go through, they also have a process of positive selection where they have to be able to interact with class 1 MHC. All right, so that was self-tolerance in a nutshell. Now we'll talk about immunological memory. And immunological memory the question is, is there innate memory? We know that the adaptive immune system has a memory. Well, technically, yes, innate immune system also has some memory, but it is for a broad class of molecular patterns, like toll-like receptors. They have a memory of what they are able to interact with, what kind of pattern they are looking for. Natural killer cells, they have a little bit of memory where they can actually have more quicker and powerful response on subsequent invasion, but it is nothing like the adaptive immune memory that we think about with immunological memory. And this trained memory is short-lived compared to the adaptive immune system. It's local and it's driven by epigenetic modifications. Let's talk about adaptive immune system memory. So for B cell memory to develop, it needs help from T cell. Either the B cells, when they are activated, become short-lived plasma cells that help in the immune response. But they can also become long-lived plasma cells. They go to the bone marrow and continue to produce small amounts of antibody, and that's one type of B cell memory. On the other hand, there can be central memory B cells, where the activated B cells become central memory B cells, live in secondary lymphoid organs, and they continue to replenish the long-lived plasma cells, or they once act again, if there is an infection, they'll make short-lived plasma cells. Similarly, there are T cells that become T cell memory cells. They are either effector memory T cells or central memory T cells. The central memory T cells live in the secondary lymphoid organ and replenish the cells when needed. And then the memory effector T cells are already in the tissue. And if there is same antigen that comes along, it can provide more efficient, quicker, and more powerful response. The properties of adaptive immune memory, we know that, say, instead of a naive T cell that is specific for one antigen, there might be 1 in a million, versus after an infection with that antigen, there might be more memory cells that would be present. It is also easier to activate the memory T cells. It needs less antigen exposure and it does not need co-stimulation. So it's easier to activate these memory T cells. And in case of B cells, there is an upgrade. So if the B cell has already switched to IgG or A or E, then when those B cells get activated again, instead of going from IgM and then switching to another antibody, it is actually a class switched memory. And then also it has better affinity, because some of these B cells, before they become memory B cells, had undergone somatic hypermutation. So the long-lived plasma cells that came from them will produce antibodies that have higher affinity. And then compared to adaptive immune system, the innate memory is not updatable, and it's same for all the humans. Compared to that, adaptive immune system is specific for each person and it's expandable. All right, talking about the intestinal immune system. Intestinal immune system has a different architecture. It has the physical barrier, which is the epithelial barrier with its tight junctions. On top of that is it has the villi that help in moving the mucus on the surface, which also helps in moving any kind of microbes in that layer. The mucosal layer has the mucus, right? And it provides that barrier, but it also has some antibacterial proteins like lysosines and defensins that help in preventing any kind of infection. And then the mucus layer will move, and that will sweep the bacteria in the lumen. There are some bacteria, in fact, there are studies that show that human body has more bacterial cells rather than human cells. So there are 100 trillion different bacteria of about 1,000 different types. So it's important that the bacteria that are in our body are good bacteria. And so the good bacteria, or the commensals, are important for a GI system, where they break down some of the carbohydrates, they help in making vitamins, and they also compete against pathogens. The important distinction between any kind of immune system elsewhere versus GI tract or intestines is that there is mostly a non-inflammatory kind of a environment. And it's important because you don't want a lot of inflammation in the place where there are other activities like digestion and absorption of those nutrients that needs to happen. So in case of inflammation, typically you would see that there are resident macrophages and dendritic cells that will carry the microbe to the draining lymph nodes, mesenteric lymph nodes, through the lymphatics. And there they'll come across, express those antigens and present those antigens to T cells. The T cells will get activated, and they will actually help in recruiting neutrophils for causing that inflammation. But most of the times there are also non-inflammatory macrophages in this area that do not produce inflammatory cytokines, and they actually, even when they are activated, they will not give rise to inflammation, and that's important. There's a lot of IgA that's produced in the lamina propria of the intestine, and these B cells are kind of prone to switch to IgA production. That can happen even without T helper cell help. This is driven by retinoic acid, and the retinoic acid is produced by intestinal dendritic cells. The IgA, what does it do? It's secretory IgA. It helps in binding luminal bacteria. It binds the bacteria that breach the barrier and pull them out, and that bacteria is swept into the mucus, and that helps in making sure that there is no inflammation. Now, some properties of the intestinal immune system include the distributed response, which means the immune cells are not only providing immunity at the site of the breach, but also along the rest of the intestine. However, even though it's distributed across the intestine, it's a private response, which means the activated T and B cells, they are only present in that area of intestine. They don't get into the systemic circulation. And then the anti-inflammatory environment that we talked about. And why is that? There are inducible Tregs that produce TGF-beta and IL-10, which are anti-inflammatory. Commensals, how do these bacteria that are going to be helpful to our body? They produce butyrate, and butyrate helps to convert the T helper zero cells. They induce them to become iTregs. There is polysaccharide A that is produced by B. fragilis microbe, and that helps the T cells in producing IL-10 and interacting with and having that anti-inflammatory cytokine available. Bifidobacterium breve, that's also another bacteria. It induces the dendritic cells to produce IL-10 as well. What happens? So we talked about how it should be anti-inflammatory environment in the intestine, but what happens if there is a pathogen? At rest, the ITregs are going to be present, but when and they are going to produce TGF beta and IL-10. But if there is a microbe, there is an environment of TGF beta and IL-6, and in presence of those, helper T cells become Th17 cells and produce IL-17, IL-21, and attract neutrophils. If there is a viral infection, for example, there might be Th1 cells that produce interferon gamma and class switch to IgG as well. So again, it depends on what kind of infection is going on. That will determine what kind of T helper cells become active. And then the last part, immune system gone wrong. We know that the immune system can sometimes still, despite all these counter actions in place, it can go wrong and cause allergies, for example, that's caused by presence of IgE that can bind to mast cells, basophils, or presence of eosinophils. And there are a lot of theories for what causes allergy, like hygiene hypothesis, maybe some genetic, genetic factors that might be also playing a role. Also, there can be autoimmunity when there is a break in the tolerance. That could be also from molecular mimicry or genetic factors that may be playing a role. And a lot of these disorders that I mentioned have autoimmune basis to them. All right, so that was the last chapter. Any questions at this?